program seminar of, thank you, robot. Yes, to the first CPEP seminar of the semester, and one of our first, one of our first virtual seminars, maybe our first, but I'm Ankur Desai, for those who don't know who I am. I am the CPEP professor here in Center for Climate Research. We feature a variety of seminars through the semester on all things climate, from paleo to modern to future, including all of the ways the climate system works together, which is one thing that we'll hear about today. And we have, we're doing 4 of these virtual seminars this semester. We've kind of cut down the number just from our usual weekly to keep it a little bit more manageable, as well as to highlight the speakers that we are bringing in who have graciously agreed to do this regardless of what time zone or part of the planet they're in. So it is my pleasure to introduce Ashley Matheny, who is an assistant professor at the University of Texas in Austin. I think I first met Ashley probably about 4 or 5 years ago, maybe longer than that now, when she was working on a paper trying to understand the diurnal cycle of water vapor. And it was, it sounded water vapor fluxes, and it sounds like a very simple question, but it turns out it's actually quite subtle and complex, especially once you start trying to compare data to theory. And it wasn't too long after that I had an opportunity to meet her in person a few times at AMS meetings. And I think the first time I was maybe a judge for one of her student presentations and was just blown away by the quality of the amazing talks that she gives and her deep knowledge of plant water use, things that I have always wanted to learn more about. So I'm always learning something new from these talks, and I hope all of you learned something new today. Just from a CV background, Ashley did do her bachelor's and PhD at Ohio State, and here at the University of Wisconsin, we will not hold that against her. She did work, she has a bachelor's in Spanish and in civil engineering, and then she did her master's and PhD in civil engineering, and she worked, her advisor is a good colleague of mine, Gil Borer, who has also given us this seminar series 3-4 years ago. Since that time, she was a postdoc at Ohio State, working on a variety of projects in integrating land and atmosphere interactions, especially with the water cycle in plants. Lots of field work, lots of modeling work, and not surprisingly, not too much long after that, got this faculty position at Texas, where you've been 3 or 4 years. I can't keep track.
This is the beginning of year number 4.
Year 4, wow, okay.
I'm
so with that, I'll stop rambling. The title of the talk today is Incorporating New Knowledge of Plant Water Use Strategies and Land Atmosphere Models, Novel Challenges and Approaches from Plant to Regional Scales. Please mute if you haven't already, and feel free to ask questions in the chat, which I can help raise to Ashley at the end of the talk, and then I'll also moderate questions by you can raise your hand. And if you really want to go into a deep dive into this, Ashley and I and my lab are meeting later on this afternoon at 2 o'clock. And if you would like to come and hang out in our lab meeting and ask more questions, just email me and I'll send you the link. All right, thanks. Go for it.
Wow, thank you so much, Ankur. I am absolutely honored to be with you guys today. Being part of your seminar series, especially with the reduced number of speakers, is cool. But thank you so much. Like Ankur said, my name is Ashley. I've been a professor at Texas for 4 years. And oddly enough, coming from an all engineering background, I'm now a professor in the Department of Geological Sciences, which makes it completely logical that what I'm going to talk to you guys about today is trees, right? So every time I do this, I always joke that I am the engineer that teaches photosynthesis to geologists. And that's sort of what I do. But let's start with why I do that thing. And so really why I do that thing is because we need it to live. So whenever plants are doing their thing about taking CO2 in from the atmosphere, not only are our terrestrial ecosystems providing the world's largest terrestrial carbon sink, but they're also, a lot of times, being the largest component of the terrestrial hydrologic cycle and the amount of water that they release to the atmosphere. And so we need both of those things. We need plants because we need them to photosynthesize so we can eat them. And we need water so that we can drink that to live. So the reason I do this is because it keeps us alive. But it's important to understand how these processes work. And it's especially interesting because those dynamics of being part of the carbon cycle and being part of the water cycle are linked together through the very same teeny tiny opening on the underside of plants' leaves, which are stomata, which you probably haven't heard about since 5th grade biology. And, you know, those stomata are used to actively regulate the exchange of water and carbon, because plants don't really want to just be giving us water all the time. What they want is they want to be taking in CO2, and releasing water just happens to be an unfortunate byproduct for them, but really good for us. And so when we talk about the dynamics of what plants do, we're interested in carbon and water exchange, but we're also interested in the way they couple the land to the atmosphere. So as our plants are working, they're moving water from deep down in the earth occasionally, not just from the soil, but from deep fractured bedrock. And they're moving that into the atmosphere, and that re-wets the atmosphere, which gives us precipitation later. But at the same time they do that, they're releasing exudates from their roots that are breaking up the rocks and creating new soil. And so there are all different feedbacks that our vegetation are providing to us that help shape the way the world works. And another one of the ones that might not be so inherently obvious is the role they play in the energy cycle. So in terms of Earth's energy cycle, all of the incoming solar radiation gets broken down into heat that will heat up the ground surface, or ground heat flux, heat that heats up the air, which that makes the most sense. So we're going to call that sensible heat flux. And then the heat that evaporates water, which is latent heat flux. And so our latent heat flux from the Earth's energy cycle, a lot of times is going to be governed by what sort of vegetation you have on the surface of the Earth, and the way that vegetation is regulating its stomata to take up carbon and release water. And so all of these things sort of interact together in a dynamic way that we need to understand if we want to be able to predict climate change, if we want to be able to predict the weather, and if we want to be able to think about whether the energy that's coming in is going to become heat or whether that's going to become evaporation. And so there are lots of natural processes like climate change and natural disturbances that will shape what the vegetation on the Earth's surface looks like. But there are also things that we do as humans, not just, anthropogenic climate change, but population growth and land use and land cover change. Like I'm showing you the increase in Austin's population because there continue to be more Californians moving to Texas all the time, that all of these things will shape the way the Earth is covered, of what we're covered in, whether we're transitioning from being a semi-arid forest, like what this part of Texas is normally covered in, to being a completely pervious concrete surface covered in condos. These are going to change what we think about in terms of how much sensible and latent heat flux we produce. So the dynamics of humans coupled with what vegetation wants to do produces really interesting results for how we think about the Earth and our weather and climate moving forward. And so usually when we think about plants and the way they move water, it's a little hard it was particularly hard for me as a structural engineer to conceptualize how much water can wood actually move, right? Nobody wants to drink from a drinking straw made of wood. That's almost impossible. But if we think about it, the amount of water that's being transpired by forests like the Amazon rainforest every day is actually more than the amount of water that the Amazon River dumps into the ocean. So this is a massive flux. And one of the things that really helped me conceptualize how much water was flowing was once upon a time when I was doing field work as a graduate student in northern Michigan, I took a video of installing a sensor in a tree that just started leaking. And so, that's not the animation I wanted to play. This is the animation I want to play. And so what you're looking at here is a birch tree, and that blue thing that's wrapped around it is an Ethernet cable. And so you can see the water is starting to leak out of this tree as I'm installing a sensor to measure the velocity of water flow. So our fluxes of water are surprisingly more than what you would think of if you just think about, okay, there's some water that's moving through wood and it moves pretty slowly. So this is shaky, not because it's sped up or anything. This is a real-time installation, but it's shaky because I'm shivering in northern Michigan in the early spring. And so as we think about that water and why and how it's moving, most plant physiologists would show you something similar to this, where our water is being taken up by the roots, it's being transported through the xylem system, and it's being released from the leaves into the atmosphere via those stomata, or those little pores on the underside of the leaf down here. And so as that's happening, we have a water supply to the plant, which is our soil water content, and we have a water demand from the atmosphere, which is our atmospheric vapor demand or vapor pressure deficit. And that's the difference between how much water the air could hold and what it's actually holding. And so this relationship between, the water moving up through the plant in response to the supply and demand function of how much water is available in the soil and how much water in the atmosphere is a dynamic process that turns out to be pretty interesting, because those two forcings, the supply and the demand, are operating on different time scales. So if you think about atmospheric demand for moisture as a function of temperature and humidity, which is something that my physical hydrology class is learning about right now, we're talking about the Clausius-Clapeyron equation, is that, that vapor demand changes on the order of half hours to hours, pretty much as your temperature changes. But when you think about the water supply in the soil, things are changing much more slowly because it's harder to evaporate water from the soil. You have to deal with the tortuosity of moving the water molecules between the pores in the soil. And so our soil water supply changes at time scales from hours, not just from hours, but from days to weeks, where our atmospheric demand changes much faster. And so there's been a lot of effort, especially within the last 4 years, looking at the way different ecosystems respond to pressure from soil water limitation versus pressure from atmospheric vapor limitation. And so my favorite paper out of this big group of papers came from Kim Novick, who's at Indiana University. And what she was looking at which one of these, the supply or the demand, is more controlling of what plants do based on what the ecosystem type is and how dry that ecosystem is. And what she found was that vapor pressure deficit, that atmospheric demand function, exerts more control in ecosystems that are larger rather than ecosystems that are dominated by crops or grasses. And so this is something that's particularly interesting to me because, like Ankur said at the beginning of the talk, I've got 1 foot in model world and 1 foot in reality. And so today we're going to sort of talk about both and how they go together in terms of both theory and practice. And the question that this really raised in my mind was, okay, so if we have some ecosystems that are more dependent on what the atmosphere does and some ecosystems that are more dependent on the soil does, how does that turn out for both the process of what's governing that response and also how we simulate it? So those are the new theories and advancements that my title hinted at today. So usually, we think about the water moving in the plant as that column of water that's moving from the soil being pulled up into the atmosphere. But I want to ask you, at least for the next duration of this talk, to think about it a little bit differently. So if we think about a plant and what it wants to do across the course of the day, and we think about it starting at before the sun comes up, the plant is completely at hydrostatic equilibrium, hopefully, with the soil water content, meaning that it's had all night with no sunshine for photosynthesis to have its stomata closed and fill up its xylem vessels so it's as hydrated as it can be. And so it starts out the morning when the sun rises completely hydrated or completely blue in this graphic. But then as transpiration starts to happen and water molecules start to be pulled out of those leaves, because of the vapor pressure gradient from the air, we start to experience a little bit of stress. And so when I click play on this animation, you're going to start to see it get a little yellow and a little red as the water becomes depleted. And as that process of transpiration goes on and water keeps being pulled from that plant, you'll see it get progressively more stressed. And then in the evening, when the sun goes down again, the plant will start to rehydrate because it won't be photosynthesizing anymore or as quickly, and it will get blue again. So in the morning, we start out blue, and then we experience water stress, and we turn a little bit red, starting at those far ends, and then we start to rehydrate in the evening. So as the course of the day goes on, we experience stress and then we relax. And so these are due to changes in water content and water potential that are going to drive water flow through the plant. But it's not very often that we actually measure water content in plants. So one of the things that I did as a postdoc as I was getting ready to leave Ohio State was I sort of started tinkering with soil water sensors and found a way to install soil water content sensors into trees with some advanced calibration. But basically what we were able to do was we were able to use these sensors, which are frequency domain reflectometry sensors that integrate the water content over a length of the time that gets inserted into the wood. And we're able to look at not just the traditional measurements of sap flow, of how quickly water moves, but we can actually look at how much water is present at any given time, which isn't something we've been able to do before. So because this was something new, the first thing I wanted to look at was, okay, if we're talking about water capacity or how much water we're holding, the traditional way that we normally would look at that are using things that look like this, that are stem dendrometers that measure micron scale expansion and contraction of the stem as it fills up with water at night and then loses water during the day. And so what we found was that there's a pretty significant relationship between the data that we're collecting with these new capacitance sensors and the data that we can see using a traditional dendrometer measurement. So that makes us feel pretty confident of, okay, we're actually seeing something that represents reality. So before we get into looking at what those data show, I want to show you the traditional data that we see and sort of set the stage for this experiment. So over the course of summer in northern Michigan, which is pretty similar to Wisconsin, we get some rain, and then we have some nice dry periods where it's fun to go out and go hike, go swim in the lake in between rainfall. And generally those interstorm periods that are dry are about 2 weeks long. And we have really sandy soil in northern Michigan. It's almost beach sand. And so that means our soil water gets low whenever we have these periods of no rainfall. And that's because all of the water is just draining out of the bottom of our soil column. And so as we're watching what the plants are doing, and we traditionally watch this via sap flow, which is how quickly the water is moving. And through the conservation of mass, we often use that as the best approximation for transpiration because, you know, you're not losing or creating or destroying water anywhere in that process. And so while we were watching our forest in Michigan respond to these dry interstorm periods, we noticed some differences between how the different species react. And so this is for red oak and red maple, which you guys have those same species near you in Wisconsin. And unless you're really good at paying attention to what leaf shape is, those trees might look the same to you. They get about to be the same height, the same width. They're both cold deciduous species, meaning that they have broad leaves and they drop them in the winter. So they might be getting ready to turn colors right now. But what we saw was that our oak trees didn't really respond to decreases in soil water content. So if you can see my cursor, this dark blue line in the bottom figure doesn't really follow the cyan line in the top. So our oak trees, they don't really get water stressed until it gets to be very dry. But the maple trees are really sensitive to what the soil water content does. And so they tend to decrease their transpiration linearly as the soil water content declines. So these are the traditional measurements of sap flow and how we normally conceptualize changes in transpiration. And what I wanted to know was, all right, so do we see changes in water content that go along with these changes in water motion? And so if we look at the data that are from those new sensors, from those capacitance sensors, we see something that looks pretty similar to what we just saw on the last slide, where every day we have our diurnal fluctuation of sort of, if you're thinking back to that blue animated Christmas tree, where we have our highest hydration status at night, and then the water goes on throughout the day, and you get these troughs as transpiration goes on. And then as you refill the next evening, you get another peak. And so we expect these diurnal cycles of different amplitude to happen. And so when we look at the storage data, we see that our maple tree is using its capacitance, and that the capacity of water that's being held in that tree is declining linearly with soil water content in the same way that its transpiration or sap flow on the previous slide declined, and our oak tree really isn't changing that much. So that would be the blue line up top. But what's curious is if you look at the amplitude of the signals. So during the day, we have our maximum amount of water storage. And so that would be right as the sun is coming up before transpiration has happened. And we have the trough in the middle of the day whenever we've depleted as much storage as we can. And so this amount of water, this change in the amplitude, is our withdrawal from storage. And so we had noticed that the amplitude in maple was much more variable than the amplitude of this change in oak. And specifically, they changed differently. So our maple has the biggest fluctuations in amplitude whenever the soil water content is sort of intermediary. But our oak doesn't really show big fluctuations in water stored until it gets to be pretty dry. So we're going to talk about that for a second. So if we talk about withdrawal, or how much water the plant is using out of that storage reservoir to transpire during the day, so we can relate the amount of water that's withdrawn to the integrated sap flux over the course of the day, we find that our maple is withdrawing water from its internal capacitance in order to be able to supply that transpiration stream during both wet and dry periods. So our maple trees are heavily reliant on storage in order to be able to function for transpiration. So they're using that water that's stored inside of their xylem system as a buffer. Now, we can plot that again, this time changing our x-axis to be soil water potential, which is basically just a fancy way of soil water content that's more related to how easy it is for the water to get sucked out of the soil by the plant. And what we see is that as we get drier, so as our soil water content becomes more negative going towards the left, we have a very strong correlation with how much water is being withdrawn. And that correlation is positive here. So what that means is that during dry periods, our maple isn't able to extract enough water from the soil to be able to transpire. And so as it gets dry, the amount that it's able to withdraw from storage to transpire gets smaller and smaller. And so our amount of water that we're using to transpire sort of is going to change with how much water is available, which makes sense. Now, if we look at this from the oak perspective, we have a kind of different story. So remember, our oak trees didn't really relate to what the soil water was doing. And before we really get into this plot, because now, there are different colors that just showed up, let's take a digression and talk about the physiology of what a tree looks like on the inside. So if you take a cross section of a tree, it looks something like this, where on the outside, you've got your bark and your inner bark, and then you have your sapwood, and that's the xylem. That's the area where water is actively flowing. And then you have the heartwood, which is sapwood that's been filled in by cellulose. That's now going to be your structural support of the tree. And then your pith is the very center of the tree. And so when we measured the oaks, one of the things I was thinking about as I designed this experiment was, so I have a sensor that's a certain length, and I want to be able to measure the water content of how much water is being moved in the water stream. And so this is a picture of the sensor down here on the bottom left. And what I knew about my oak trees was that oak trees are notorious for having a really narrow band of that sapwood, or that area that's actively pumping the water. And so one of the things that I wanted to do was I wanted to be able to compare the oak and the maple, apples to apples, right? But maple have big, thick sapwood. And so if I put the sensor into the maple, all of those metal tines, the entire length of the tine, is embedded within a hydroactive xylem, so material that's actively moving water. But if I were to put that whole length sensor into the oak tree, who has narrower sapwood, it would be integrating over not just the sapwood, but it'd also be integrating over part of the heartwood that's not actively conducting. And so fortunately for me, I should never be left alone with a rotary tool. The sensors have solid steel tines, which means you can cut through them. You have to use a different calibration equation that you have to develop for yourself, but that's okay. That's a surmountable problem. And so in order to make these measurements in the oak tree, to compare them apples to apples with the maple tree, I cut my sensor. And so I put a long sensor that was not cut, so the full length of the tines, right next to a short sensor that was specifically cut to only be in that hydroactive portion. And so that's where all of these different colors come up. So on this figure, we're still looking at how much of the storage withdrawal we're using to supply the transpiration stream. But now we're looking at it in terms of wet and dry and shallow and deep. So the short sensor that's just in the hydroactive xylem during wet periods is represented by blue dots. During dry periods, it's represented by wet red dots. That long sensor that integrates over all of the wood, so not just the hydroactive portion, but the heartwood as well, are the cyan and the magenta, where cyan is wet and magenta is dry. And so basically the point I wanted you to see here is that we only really have a relationship for oak using the water withdrawn from storage when it's wet, and only in the outermost band. And if we start to look at that more in depth, in terms of how that were to change with soil water potential, we see a really interesting story start to emerge. That just like our maple, we don't have a relationship between how much water we're taking out of our hydraulic capacitance when the soil is wet over here on the blue and cyan dots. But we do see a strong relationship start to emerge of how much water we're withdrawing from storage when the soil gets to be dry. And the interesting point here is not just that the dots are magenta instead of red, but also that the relationship, the direction of the correlation, is reversed from what the maple was doing. So remember, the maple amplitude started to shrink down and get really little as it got dry. Now our oak amplitude of the diurnal withdrawal from storage started to get bigger when the soil got dry. And that's what you're seeing here. And so our magenta line in this case is that withdrawal from storage, but this is the full length sensor that's penetrating not just the part of the wood that's actively conducting, but also the wood that's being partially infilled by cellulose. And so my hypothesis, and I'm working with a collaborator who is a genius in instrumentation design, to build a sensor that we are able to test this, but my hypothesis is that partially infilled wood is still porous enough that it's able to hold water, but whenever the oaks start to get stressed out, that water can be sucked out because the vessels are laterally connected, and it can act as a battery backup system for transpiration that helps our oaks not be as stressed as they would be otherwise. And so this is one of our interesting findings of, we should go, and we're going to look a little bit more at this in the future. But we can look at this from another way too. So instead of looking at just that dynamic of what is a plant doing within one day, we can look at what changes between one day and the next. So this is the figure that you've seen before. And we were just talking about that concept of withdrawal. But now instead of looking at the peak to the trough, we're going to look at the difference from one peak to the next. And we're going to call this depletion. So this is the difference between how much we had stored at dawn on day one and how much we were able to store at dawn on day two. And the difference between those two is just related to how much water that the tree can extract from the soil. So as our soil is declining, our maple storage is becoming depleted because we're not able to replenish it all the way because there's not enough water being supplied from the soil water supply. And so if we were to look at this metric of depletion for both the oak and maple, what we would see is kind of similar to that first time we looked at the maple, when we saw really different behaviors when the soil is wet versus when the soil is dry. So when the soil is wet, we don't have a relationship over here on the right. But as our soil dries out, we have a strong relationship of the depletion of stored water and soil water potential. And so one of my other hypotheses here is that our maple tree is exhibiting this really clear threshold response in terms of what its water content is that tells us exactly when the maple starts to get stressed out. This is the point when I can no longer withdraw as much water from the soil as I need to be able to buffer my transpiration stream during the day. And this is a really interesting thing, is if we're able to simulate this, or if, say, we were able to see this from space, we could be able to tell exactly the point at when plants become water stressed, which would allow us to predict things like the onset of drought or the water content that exists inside of potentially fuel for forest fires. And so this has a lot of really neat implications that we're still exploring. So in terms of having 1 foot in reality and 1 foot in models, let's switch gears for a little bit and let's talk more about those models of, can we use models to predict this sort of threshold response of how do we look at onset of drought? So in order to do that, the first thing you should know is, how do current models see vegetation? So if you're talking about a land atmosphere model, which are the models that go into global climate models that do everything about the interactions between how the land and the atmosphere work together, those models see plants as what we call plant functional types, or PFTs, which is basically just a way of grouping plants together of, okay, you all live in the same place and you look the same way and you do the same thing with your leaves, so we're going to put you in the same category. And so most land surface models group all of the plants on Earth into about 15 different groups. Some models use as few as 8 groups and others use as many as 40, but in general about 15. And so if we're thinking about those plant groups, within a plant group, everything is done on the basis of saying, all right, So you look pretty similar, and you probably function pretty similar. And so we're going to say that you have the same leaf, or the same sun leaf and the same shade leaf. And so if you have 15 plant functional types and each one gets a sun leaf and a shade leaf, you've got 30 leaves for a global climate model, which might not be the best option, particularly because if you're looking at just leaf habit of are you deciduous or evergreen, or leaf shape of are you a needle leaf or are you a broadleaf tree, our oaks and our maples that we've been talking about up until this point, those are both deciduous broadleaf trees, which means that if we're a climate model, those are the same tree. But more importantly, those aren't just the same tree, those are the same leaf. So we don't really have a way to simulate the way different plants operate within climate models. And so one of the first projects that I ever worked on as a master's student with Paul and Ankur as some new mentors for me was looking at the North American Carbon Program data. And I was asking the question of, okay, how well do models do at predicting latent heat exchange or transpiration? And I was really hoping that within my master's project, I would see that there was one great model formulation that was always the best, or one ecosystem where models excelled and one ecosystem where they could improve. But I didn't find any of that. And so what I found was that models do really well on long time scales. And overall, things come out pretty evenly. There aren't winners or losers in terms of ecosystems or winners and losers in terms of formulations. But what was interesting that came out of this was we did find an error pattern. And so if you're talking about models, you often talk about model error, right? Which is the difference between what the model simulates and what we observe as reality. And so if your model error is a constant overestimation or a constant underestimation, you have a systematic bias and you can adjust for that. Or if it's equally over and underestimating, you have a random error, which is sort of expected. But if you have a consistent pattern of error, you can learn something about a process your model is missing. And so the error pattern that we found was that our models for different ecosystems and different model formulations were underestimating transpiration in the morning and compensating for that by overestimating transpiration in the afternoon, which led to a net zero error or close to zero error on the order of a day. But we are missing the dynamics of what those plants are actually experiencing and how they're regulating their stomata. So if we go back to that data that we were collecting of the way plants actually work, one of the things we can see is that when plants are transpiring over the course of the day, and our x-axis in this figure is going to be time of day, and so these are averaged out. And so on average, in the morning when the sun comes up around 6:30, things start to pick up, and then they go and they go and they go pretty quickly up until solar noon, which in Michigan is about 12:30, 1 o'clock. And then they start to slowly decline. So it's not a perfect parabola of the sun, but because plants experience that stress, they start to slow their transpiration down a little bit, which flattens the curve on this side. And flattening the curve has become a big thing lately. And so this different asymmetry in the way that plants control transpiration looks a little bit like that error pattern we saw. So if I bring that error pattern back up, and you look at the time of day and you think about that overestimation or underestimation of flux in the morning, followed by the overestimation of flux in the afternoon, and you squint and you tip your head to the side and you sort of pretend, for the sake of me right now, that you can see that that underestimation of flux lines up with when plants are really doing the thing in the morning, and that overestimation of flux lines up with whenever plants have slowed down because they're stressed out in the afternoon. And so in terms of our models, we're not capturing the dynamics of transpiration. We're not able to capture how plants are responding to vapor pressure deficit or even to soil moisture. And so if we keep trekking on through model world, and because I'm an engineer, let's get to the math of things, of most land surface models represent plants like this. And this figure is from a paper from 1993, which I know, super old. Is plants are simulating, or models are simulating plants as some sort of resistance to water flux, right? Where our stomatal conductance is going to resist the exchange of water into the atmosphere and resist carbon coming into the plant. And that function that dictates what our stomatal conductance is going to be, can look something like this guy, which is the Looning model from 1995. And we're basically saying that our plants in this equation, which don't worry about what the variables are, but I'll tell you that this stomatal conductance formulation is regulating what the plants are doing at their leaves on the basis of photosynthesis, because plants want to be taking up that carbon, right? And vapor pressure deficit. And so there's something missing here, that even though Stenard's diagram of what the model leaf looks like has a stem and a trunk, of there's nothing over here that says anything about what the soil is doing. And there's certainly nothing over there that says anything about what the inside of the plant is doing, about how we're releasing or taking up water from our storage to withdraw water from that reservoir. So how do models do soil? So models do soil as pretty much a binary thing, where you have a function that says that if stomata are closed, so if the soil is really dry, like at the wilting point, stomata are shut and you have a value of 0. Or if the soil is close to saturation and you are well hydrated, you have a value of 1, which means your stomata are completely open and things are great. And then somewhere in between, they can be connected, that 0 and 1, by a linear function or a sigmoidal function. But there's nothing in between, no way to communicate exactly, what the roots experience to what the leaves experience, which from physiology, we know that there's that whole system of vasculature that is dictating the way water is moving. And we're using that to supply storage and to be able to buffer that transpiration stream. So within models, there's really no way of capturing how plants behave in an accurate manner. So recently, and I say recently within the past 3 years, there have become a lot of new models that we call plant hydraulics models that are super cool in that they're starting to put in different ways to simulate what goes on in between the soil and the leaf. And so now, instead of just seeing that leaf as a resistor to flow, we've got a bunch of resistors to flow, where we have resistance in the root. So we've got this process of this plant taking up water from the soil. We have resistance in the stem and capacitance in the stem, where we're able to withdraw water from a reservoir and then pump it to the top of the tree. And so there is this new opportunity of advanced plant hydraulics capable models to simulate the way vegetation are actually functioning. And so this paper is under review in James right now, and it is from one of my PhD students, Ling Ching, who just took a position as a postdoc at the Pacific Northwest National Lab. And so he put one of these plant hydraulic schemes into the Noah-MP model. And Noah-MP is used for weather predictions most often, but is also a climate model. And so within Noah-MP, the first thing that Ling Ching and I asked was, all right, can you simulate those two behaviors of oaks and maples, of who uses water from storage when? And so what Ling Ching found was that using his model, he was able to get a pretty okay approximation of what the plants were really doing. So in this figure, blue is oak and red is maple. And our observed data are dotted lines, and our modeled data are solid lines. And so we can see that we've got the amplitude signal going on of each day the plant is withdrawing some water from storage, and each evening it's replenishing that storage. And we can see that the maple is declining with soil water content, but it's not perfect. And so we're doing okay, but there's still some room for improvement here. And one of the things that Ling Ching was most frustrated about that really excites me is looking at the difference between how we are responding once we start to input water to the soil again. So if we keep going past this dry down and we look instead right here, on just before day of year 225, we added some precipitation. So our soil water jumped up, and that made that linear function of 0 to 1 for stomatal conductance change, right? So at that point, 100% instantaneously, the plant became totally rehydrated and was happy and able to function again. But in reality, in those dotted lines, what we see in our observations is that it takes maple, the red line, a lot longer to start responding to increases in soil water content, because it has to refill all of those xylem vessels that got emptied out when it became so stressed it was using all of its water storage reserves to be able to transpire. And so this is a new opportunity for advancement in terms of that plant hydraulics community, where we can start to develop advanced models based on hydrology that will allow us to simulate what goes on inside of the vascular system. And so we have this other model that's called the FETCH model, which stands for Finite Element Tree Crown Hydrodynamics. And I'm not going to say that again, but what you should know about this model is that within this model, we're using the same sort of finite element, finite difference structure that you would if you were typically a groundwater hydrologist and you were simulating water flow in soil. Is that we're approximating that our xylem is a porous media. And we're saying that that's an unsaturated porous media, which is an important part. So when we as hydrologists simulate unsaturated porous media flow, we use the Richards equation. And so our Richards equation, if you look at it as a hydrologist, usually looks something like this, which I know at this point is just eye candy. Don't worry, this won't be on the test. But we can rewrite that and we can expand it out so that it looks something more like what we see with the dynamics of water transport in the plant. So we have a modified Richards equation where we've got a capacitance term, and our capacitance is going to change as a function of what the water potential or the water content is going to be. We have our conductance term that changes based on what the xylem looks like. So our oak have big vessels, but very few of them. Our maple have a lot of really small vessels, and pines, well, they use tracheas instead of vessels. And so using these parameters or using these functions in the Richards equation, we can actually capture those different physical properties of the wood when we create numerical simulations. And then of course we have our pressure gradient that will change based on how tall or short the tree is, and transpiration. So when we use the Richards equation and we're simulating water flow through a tree, we have the opportunity to parameterize it based on observation. And so we can use different parameter sets within this model to simulate the way stomata respond, whether they prefer to be more conservative and shut so that they're not losing too much water like our maple tree in red does, or whether they like to leave their stomata open because they really want that carbon like our oak tree does in blue. And we can parameterize how conductive or not conductive the xylem is based on what the vessel size and the vessel distribution are. And so using this model that relies on the fundamentals of hydraulics, what we're able to do is we're able to simulate our decrease in maple transpiration and kind of that constant in the oak transpiration, which is neat, but is it enough, right? Because right now we're just thinking about two trees. And when we started this digression into model world, I told you that these two trees are part of that same plant functional type. And so if we actually wanted to implement something as crazy as the FETCH model into a land atmosphere model, we'd really have to think about what are the logistics about making that happen, right? So we're simulating individual trees, when if we want to be a land surface model, we got to be simulating thousands of trees. And so one way to scale is going from what we see in the land to the top, right? And so if we wanted to do that, we can use some remote sensing data to help us talk about what the whole forest does instead of just what individual trees do. And so we can use multispectral imagery to break down our forest into what's the distribution of species that we have. And so each species is slightly different colors and different wavelengths. And so we can break that apart, and we can use aerial LiDAR to get height, which allows us to break down our whole forest into bins of species and sizes. And so using those histograms, we can just say that, okay, we assume that one medium-sized maple tree is going to perform pretty similarly to what the next medium-sized maple tree does. Or we can say that one large oak is going to transpire and use its storage in the same way as the next large oak. And we've done this with our measurements of sap flow, and we've compared that to some eddy covariance data, which if you've taken Ankur's classes, you probably know what that is. But basically, we see some good results at scaling from the ground to the top. But now, as we start to think about big ecosystem level models, the question we have to ask is, can we go from the top down? And so if we look at remote sensing data, and this paper is from Alex Konings, and it was published in Global Change Biology. She's using satellite data from the AMSRE satellite, looking at a metric called vegetation optical depth, which is sort of a proxy for how you can sense water content from space. And so those data that we were looking at and we talked so much about in the beginning of the talk, about the capacitance and the water content that's actually present in the trunk of the tree and how it changes, this is satellite data that we could potentially match up to those field-based data. And so we still have to kind of work out a few little differences in order to be able to use this. So whenever we see data from the AMS-3 satellite, that's the vegetation optical depth, that satellite data, or that remote sensing data, is going to see all the way through the leaves, and the branches, and the trunks, and even into the top 5 centimeters of soil. And it's going to integrate all of that moisture signal and say that all of that this is what vegetation optical depth is. But now that we know that we can see diurnal fluctuations and that we can trust data that are coming from sensors that go into individual trees and measure water content in wood, so meaning stems and branches, and we have lots of armies of undergraduates that can go out and measure leaf water potential, and so we can know what the water content in the canopy is. Now we can start taking apart that remote sensing data and breaking it down into, this is the water content that's in the leaves. This is the water content that's in the wood. And this is the way that water content is changing based on what our ecosystem is made up of. And this presents what to me is one of the coolest opportunities, because now, rather than just having our individual measurements of, Ashley goes out and she measures something in the forest, and Ankur goes out in a forest in Wisconsin and he measures something that, is on the same basis, but they get different results. Now we can look at this in terms of, okay, we have one data set that's a remote sensing data set. So this is a size that can be imported into a land atmosphere model on a large scale that we can relate to what we see in Ashley's site and in Ankur's site to understand more about what that data is telling us. And we can use that in conjunction with these advanced physics models like Fetch2 or Noah-MP Hydro that will allow us to simulate the dynamics of what happens. And now we can potentially be able to simulate, okay, we see that threshold response of the way water content is changing with soil moisture. That lets us tell the onset of drought for this ecosystem. Or we can tell we have reached a critical threshold of canopy moisture, which that allows us to predict the likelihood that a forest fire will ignite and become huge. So there are lots of big opportunities and ways that we can start to see and use these data in ways that we haven't ever been able to before. So that is the last slide that I really want to show you of the implications of all of this stuff, is that our water content in biomass is actually going to play a critical role in supplying water for transpiration. And that's something that we haven't really had the capacity to measure before. And what we're learning is that not all species are using that stored water the same way. So our red maple relies on it every day for transpiration. Our red oak really only relied on it when the soil got really dry. And right now, we don't really have a way to simulate those two discrepancies. And for some species, what we're seeing is that this could reveal a threshold of this is a drought onset point. And now we can tell that in a way that we weren't able to before. And so our new plant hydro models are capable of representing these dynamics, but simulating their responses is still a challenge, especially in terms of the response to re-wetting the soil. So remember in Ling Ching's figure that he saw that really sharp increase of, okay, the soil is wet, here we go. But our plants in reality, they don't do that. And so this is a new opportunity for places where we can use advanced physics to be able to simulate the dynamics of how that happens. And the Richards equation is one of those big possibilities. So I want to thank all of my collaborators who have helped me do all of this work. This is Dr. Liang Yang, who works with me as a colleague at the University of Texas. And Liang and I have co-supervised Ling Ching, who built the Noah-MP model. And then these are my graduate students, my PhD candidate, Anna Maria, who is an instrumentation whiz, who has taken care of keeping all of our sensors up and running for most of the past few years, although she's gotten a little break because of COVID this summer, and my master's student, Austin. So with that, and thanking all of those guys, I want to thank you guys for your attention. And if you have questions, if you're still awake, I'm happy to answer them for you now.
All right. Thank you. That was a great tour de force of many different aspects of the water cycle. I love it. Open for questions. Feel free to either use the raise hand button and I'll unmute yourself and I call on you, or send it in a chat. And I'll wait a second here. I'll start off with a question while people think of theirs. So of course, one of the things I like to think a lot about is how do we improve these kind of global scale models? And you've brought in a few different aspects. And of course, one thing that's very straightforward to me is, right, well, in virtually every GCM land atmosphere model, maples and oaks are the same plant functional type. And you're showing all these simulations where you're doing different things with those two species. What would be your, like, if you were saying, what was your number one step if you were to run a global model? Like, where would you draw that, would you create a new plant functional type basically between isohydric versus anhydric, or is there something else that, you would think would be the biggest bang for your buck?
So there are two parts of your question, and one of them I want to address first is this difference between isohydric and anisohydric. So to give you guys some context of what Ankur is talking about, is iso is from Greek, right? And it means the same. And so plants that are isohydric like to keep their leaves the same hydration state all the time. Anisohydric plants do the opposite and allow their leaves to get really dry in favor of taking up carbon. But what we've learned from especially the work of Kim Novick and Bill Anderegg and a lot of others is that isohydry, of what we think of as the stomatal strategy, is really an integration of what we see in response to the environment and also the rest of the hydraulic system, right? Because what your leaf hydration state is going to depend on what the branch next to it is and what the trunk next to that is. And so we really are starting to see that more of, instead of like this is really a trait, is this is a response. And so maybe isohydry isn't the way to do it, but to answer the other half of your question about, being within that same plant functional type, I think what we do need to do as a modeling community is we need to recast those functional types, where currently, they are based on plant traits. They're based on, leaf habit and what the leaf shape is and where they exist biogeographically. But I think the best way to reshape those is to reshape those based on actual observable traits of, this plant has this type of xylem, or this plant has this type of root functionality, of whether it's a sinking root or whether it's a lateral root that allows it to move. But it's still like, that's one of the biggest areas that we have for improvement, is everybody who does work on plants, whether you be like me who works on the plant water cycle, or someone who works on nitrogen, wants to recast plant functional types in terms of their own specific silo. And so as a community, I think figuring out the best way to cast plant functional types, and it might even end up just depending on the question you were trying to ask of that model at that simulation run, but it's still completely open-ended of how do we redefine these in a functional way.
Yeah, very cool. Thanks. So we have a few questions coming in on chat, so I'm just going to read them. And while I'm reading, if the person wants to unmute and follow up, they can. So Samantha Chatterjee asks, thank you, first of all. Do you think soil texture could be an important factor for controlling sap flow, and could it be a bottleneck for model simulation?
Absolutely. And so when I showed soil data, especially with those correlations with what the plant was doing, that what I was showing wasn't necessarily soil water content. What I was showing was soil water potential. And that's one of the ways that we use to look at the ability, or how hard it is to get water out of that soil. So if you are one of my colleagues who likes to think about things from a geologic perspective, a lot of the times we'll talk about soil textures, and we'll talk about clays, of how clays really tightly bind water, and that 50% water content in a clay is really not that much. But 50% water content in a sand that likes to drain easily, that's a ton of water. And so when you compare it water content to water content, you're comparing apples to oranges based on the different soil textures. And so soil water potential is a way that we can talk about the water content with considering soil texture and how hard it is to extract water from those. And so absolutely, you are completely correct that soil texture plays a huge role. But fortunately, because we use water potential, we're able to sort of eliminate the effects of it.
Yeah, thanks. It always blows my mind how low the soil water content at the UMBS sandy soils are, but it makes sense when you put it in other contexts. So Kate McCullough asks, do you have any guesses about traits that oak or maple have that would allow us to predict what other species would do? Would it be related potentially to wood density or wood type?
I think wood density is going to be a super huge trait, and there have been lots of trait analyses. So there's the Global Plant Trait Database, and wood density is one of the most easily measurable traits, and they have a ton of it. And so I think that that presents this huge opportunity, because not only does that capture sort of the porosity and the number of vessels that they have, but it would also sort of allow that integration of what the overall density is, so not just sapwood, but also a little bit into the heartwood. So yes, I think wood density is going to be one of those things that comes up a lot. One of the other ones that I found a lot of papers talking about recently is vegetation height. And vegetation height to me is not quite the whole story or as informative as wood density would be, because wood density would be really controlling. And I can see tree height as controlling in terms of the hydraulic path length, but I think that there's more to the story there. So yes to wood density, but no real predictions about anything else. Maybe maximum xylem conductivity, which is a wood trait that's frequently measured. It would be one of my favorites that you could use, which helps dictate what the shape of that cavitation curve is, which is one of the things that we plug into the Richards equation when we use the Fetch2 model. But there are lots of them, and I think one of the biggest challenges that we have is finding the way that those traits covary and trade off, so that we can include more than one of them as like a trait space of, this is like a viable strategy where you have this sort of root depth coupled with this sort of xylem conductivity and that sort of thing, if that makes any sense.
Cool. All right, we have another question in from Aaron Alexander, who asks, thanks for a great talk. Have you thought about the possibility of coupling between groundwater table, vegetation, and the overlying atmosphere as a driver in the plant hydrologic cycle in any of your studies? And if so, what are the main points or difficulties that you found?
Absolutely, Aaron. So one of the most interesting points, and I think if you were to talk to Gordon Bonin at the National Center for Atmospheric Research, he would say it's one of the most frustrating points, is that when we talk about those coupled models, those land atmosphere models, we have lots of soil layers. That we have like 20 different soil layers, because the groundwater people are so influential in how those models are developed. And we have so many atmospheric layers, but we don't have vegetation layers. We get one leaf or two leaves. So I guess if you have two leaves, you can think of it as two layers. But one of the things that we are fighting for, sort of as a plant physiology and ecohydrology community, is more stuff within the plant canopy. And so as we start to think about that more stuff that goes on, one of the things that Gordon, because I said this is his pet project, is looking at is creating multi-layer canopy models. And so this would be something that looks like that Fetch2 diagram that I showed you, where it's broken down at different heights based on what the leaf density is at different points in the forest. And so in terms of that coupling, it becomes a much more interesting perspective, because you can include all of the different dynamics of what different stages in the canopy see. And this, for tropical people that have very distinct canopy levels, is really cool. And so that's absolutely one of the biggest drivers that I could see is creating change within these land surface models, is increased resolution of the way we see plants.
And there's one last question we'll ask before we send everyone off. So Karen Russ asks, how can we start to improve models in terms of storage response to rewetting?
So the first thing that we, and specifically, I mean I, because this is my active research project right now, is we need to understand how that storage responds to rewetting. So one of the cool things within plant physiology that's sort of been an argument is whether plants refill or do not refill their xylem vessels. So if you think about xylem as like the tubes that are transporting water to the top of the trees, once a vessel is stressed out, it can embolize, meaning that there's a little air pocket that forms within the xylem vessel. And before you can refill that vessel, you have to fill up that air pocket with water so flow can resume. And that process of filling that air pocket in is often energetically costly, where they have to generate a gradient of potential to push water in from another neighboring vessel to fill it up. And so some plants have been shown to refill embolized vessels and restore flow. And other plants have just been shown to completely abandon those cavitated or embolized vessels and grow new xylem. And so it's going to become a big problem of how we understand what plants do to refill their xylem vessels, of understanding what that storage response to rewetting looks like, and whether that's the same or different for all species, and if we can simulate it. So a lot of what we saw with the Richards equation was that we can use this to simulate unsaturated porous media flow in soils. And so in studies where they've been looking at Richards equation simulation for soil water content and soil water potential, is they see this hysteretic response of the way that the soil re-wets is based on what the antecedent condition was. And so it's my hypothesis that we can do the same thing for re-wetting in the xylem system with the Richards equation. But in terms of parameterizing that for how it's actually going to look based on different species, just looking at the preliminary data that I showed in this talk, that's going to be a bear of a problem. So much so that I'm begging the NSF for money to do it.
Okay, well, thank you for answering all those questions. Really great. And thanks for hanging out. And we'll talk to you again in a half an hour. So as I mentioned, if anyone wants to join our lab meeting, just let me know. In our next CPEP seminar will be next month, October 12th on Indigenous Peoples Day. We will have David Kristovich from the Illinois State Water Survey. He's a boundary layer meteorologist who works on understanding the interactions between land-atmosphere interactions, like we heard about today, with atmospheric flows. So we'll get to kind of maybe even build a little bit from what we learned today to think about how the atmosphere responds to some of these. And so that's October 12th. So stay tuned for that. And thanks for sticking around for the talk. And thank you, Ashley.
All right. Thanks a lot.
All right. Cool.